So, hello, I'm Fabien Zoulim from Lyon University, and I would like first to thank the organizers for the kind invitation to give this talk on patient selection for clinical trials based on drug class. You see here my disclosures. As you all know, the field of drug discovery has made a lot of progress for the therapy of chronic hepatitis B. And we have now many assets that belong to different classes of drugs and compounds based on their mode of action that should be considered for clinical trials. So we have drugs that interfere with the viral life cycle, that inhibit viral replication with specific targets, or can inhibit viral antigen expression. Besides these direct-acting antivirals, we have drugs that modulate the antiviral immune responses with strategies that invigorate immune responses or stimulate specifically these immune responses, as well as newer strategies aiming at replacing these immune responses. So we are at a stage where we have all these different assets, and it's very exciting because we now have to see how we are going to combine those different drugs and in which patient population. As you know, chronic HBV infection is a heterogeneous disease, and as well as it affects different patient populations. So we have different phases of the infection and disease, and drugs with different mode of action may be best suited for specific phases, but this has to be demonstrated by clinical trials, and we'll discuss this in the presentation. Patients may be at different stages of liver disease, and we need to be cautious for safety, especially with patients with advanced fibrosis. And patients may have different treatment history, as we will see later on. And they may have different routes of transmission that are usually associated with different pathobiology, different ethnicities, HBV genotype, or mutant profiles. So you all know the different phases of the chronic HBV infection that have been revisited by the EASL clinical practice guidelines. And I will not go through all these phases, but mainly they are the reflect of the interplay between viral replication, immune responses, and inflammatory responses. And we can then distinguish different phases of the disease that may not be automatically sequential, so which is also something that is very tricky in the management of the infection. Now let's see the different components that we want to discuss today. First, the stage of the disease. And this is particularly important with respect to liver flares. Obviously, with any type of drugs and mode of action, we are always afraid and should be aware of on-treatment liver toxicities, as well as other type of toxicity. We have also to take into consideration the immune-mediated flares during treatment, and this may potentially apply to all immune modulatory agents. And there are also flares that may follow treatment cessation, especially when they are linked to viral rebound and immune-mediated flares. So for taking all this into consideration, at the early stage, we should exclude patients with advanced fibrosis, and they need to be carefully assessed by all the clinical means and by expert hepatologists. And there are also some cutoff values for non-invasive measurement of liver fibrosis with a FibroScan value of less than 9 kilopascal. Just as a reminder, you all know the sad and difficult history with the combination of nucleoside analogs and inarigivir, a RIG-I agonist, which was shown to have a quite interesting effect in the first phase 2 clinical trial for 12 weeks administration. But then when the second phase 2 with a longer administration of inarigivir was performed, then several patients presented severe side effects, and one patient developed lactic acidosis with multi-organ failure and died from it. So this should be always kept in mind with any type of mode of action. Now, the phase of the disease. The phase of the disease is a reflect of the interplay between viral replication and immune responses. This has been nicely summarized in a recent review. And again, I will not go through all the details, but clearly you all know this HBe positive chronic infection, the so-called immune tolerant phase, where viral replication levels are extremely high. The adaptive immune responses are exhausted, but not as much as we thought before. And then patients enter the chronic hepatitis phase, where liver inflammation takes place, as well as fibrosis. So these are important consideration, because this may impact the way we want to evaluate the new antiviral drugs that are in development. For instance, there are a strategy where investigators try to combine direct-acting antivirals with different mode of action, nukes, capsid assembly modulators, siRNA, to see whether we can deplete the pool of cccDNA. And this is summarized here. And in that situation, I think that one ideal population for a proof of concept study, it's not for registration trial, but a proof of concept study would be a patient in the so-called immune tolerant phase, whereas the goal here would be to demonstrate that the depletion of the cccDNA reservoir can be achieved or not without an immune-mediated hepatocyte killing. So this could be extremely interesting when combining 2 or 3 modes of action. Obviously, that this does not exclude the patients in the classic chronic hepatitis B phase, where this type of combination could be applied. Now, there are other type of combinations where we combine direct-acting antivirals with different strategy to modulate the immune responses, with immune response invigoration and or immune stimulation. And with that, I mean, there are different patient population to consider, and the latest studies really suggest that young patients in the so-called immune tolerant phase would be an ideal population for proof of concept studies again. And the goal here would be to demonstrate whether a reduction of viral antigen expression is an important component of the immune reconstitution strategy prior to specific stimulation of immune responses. Obviously, we could say, well, and what about patients in chronic hepatitis B phase? There are also studies, I mean, that suggest that the chronic inflammation may be also associated with negative signals in the liver immune microenvironment that would make the specific stimulation of immune responses less efficient. So it might be important there to have a viral control prior to immune stimulation. Here is a very interesting study published by the group of Antonio Bertoletti last year, where they really look at the impact of age, level of HBs antigen on the adaptive immune responses, and they made really an extensive and comprehensive study there. And they showed that the duration of HBs exposure rather than the quantity of HBs antigen was associated with the level of anti-HBV immune responses, and that the strategy, and suggested that strategies to restore anti-HBV immune responses should be considered in patients younger than 38 years of age. So this is really interesting. And in that direction, there are new clinical trials that are starting based on strategies to knock down viral antigen expression prior to a therapeutic vaccine to stimulate specifically the immune responses. And they are based on proof of concept study in mouse models where transgenic mice that are immune tolerant were treated with siRNA followed by a therapeutic vaccine. And it was shown that in that combination, then the immune responses induced by the therapeutic vaccine were the best in terms of HBs antigen decline, HBV DNA decline, and anti-HBs seroconversion compared to a therapeutic vaccine alone. So this patient population, young age, would be really the best fit for this type of combination. Now we have to consider also the treatment history and treatment schedule. We have many patients that are virally suppressed, and in those patient population, we can evaluate add-on strategies of direct-acting antivirals to deplete the cccDNA reservoir, for instance, by adding capsid assembly modulators in nuke-suppressed patients, or to add not only CAMs, but also strategies to decrease viral antigen expression, siRNA, antisense oligonucleotide, and see whether this can enhance the effect on the cccDNA reservoir or can participate to the immune reconstitution. There are obviously strategies in those patients that are controlled, virally controlled, to add on immune modulators, TLR agonists, checkpoint inhibitors, therapeutic vaccines, where patients would be in a more favorable inflammatory environment based on the viral control. Another point to consider is whether stopping therapy should be considered only after reaching the endpoint, and there all the questions remain regarding durability of response, whatever the mode of action of the drug. And in any case, we should exclude patients with advanced fibrosis. And the other point is whether the stopping strategy should be a component of the cure strategy, and whether this would increase the rate of functional cure after treatment cessation. And you know that it is still debated when we think of only stopping nukes. So for the newer mode of action, we still don't know. And in any case, we should be extremely cautious and exclude patients with advanced fibrosis because of the risk of relapse and flares. Now, just to come back to this timeline consideration, when we stop treatment, whatever the mode of action of the new drugs, there will be a follow-up phase. And in that follow-up phase, either we would be in consolidation phase if the endpoint has been reached at the end of treatment, or if the treatment stopped even before losing HBs antigen, then the consideration is whether the flare phase may be beneficial to the patient. Now, there are another point that is very important is co-infections. And we have specific drugs, entry inhibitors like bulevirtide, which, as you know, block specifically the HBS, the envelope protein interaction with NTCP receptor on the hepatocyte membrane. And bulevirtide so blocks HBV and HDV viral entry. So for entry inhibitors, the best population would be there's a scenario where we have a high turnover of infection or a large liver space, and this is typically the case for HDV infection. This is the reason why bulevirtide is mainly developed for chronic hepatitis delta. For HBV, the best population would be patients with increased and high cell turnover, so acute hepatitis, severe hepatitis, or HBV reactivation. But to my knowledge, there is no study that has been performed yet. In virally suppressed chronic Hep B patients, bulevirtide may not be evaluated at best in this population for proof of concept because of the slow rate of new round of infection in these patients. But it could be part of future combination at later stage. So to conclude, so we have now more and more drugs entering phase 2 clinical trials with different mode of action. So we need to be really chosen in terms of patients population regarding the disease phase, disease stage, patient's history, and for some mode of action, co-infections with the Delta virus. And for this, we need to have carefully designed clinical trials to address both the drug's mode of action and patient's heterogeneity. And this may be part of large platform trials for companies that have different assets, or small targeted studies that would address different mode of action or different patient population. And with this, I would like to thank you for your attention.